The first thing is Bob Sisko. He's going to be talking about the GMP. GMP stands for Good Manufacturing Practices. Grade Ibogaine, which is Ibogaine made as a purified hydrochloride in an FDA-approved factory. In an FDA-approved factory, so that it is Ibogaine for purposes of doing clinical trials.
GMP Ibogaine. I've been working with Ibogaine in its development since 1983, and we have determined that lack of clinical studies, the paucity of data, is a result not of the lack of interest by the scientific community, because there have, after all, been hundreds of papers presented in preclinical areas, but the lack of a GMP product. And so we've directed our energies into two areas. One is manufacture of GMP Ibogaine, and the second is utilizing it in a clinical trials program. So we'll talk about that and organizing an international multi-site study evaluating the safety and efficacy of Ibogaine in the treatment of opioid withdrawal. So this is, it was actually prepared for the Indian Psychiatric Association, which I met with in Bangalore. And it relates, of course, to the development of this unique anti-addiction medication. And there are 3 corporations participating in this research and development effort. The RAND group, as it's called, Research and Development, is the Addiction Research Institute, which is based in Delaware, Humatech, which is in Israel, and Phytostan Enterprises, which is a Quebec corporation based in Montreal. So we'll talk briefly about the product, the background, the anti-addictive psychiatric uses, preclinical, clinical data, and our proposal for clinical studies in India. Well, as we all know, Ibogaine is found in the West African plant Tabernanthe iboga. It is also found in other plants all from the same genus, including Voacanja africana. These are the iboga roots, and the alkaloids are concentrated in the bark of the root. It is then scraped off the bark and prepared for consumption in Bhuti initiation rituals, and for general use in small doses as well. Total alkaloid extract is a brown clumps of brown material which contains a dozen or more alkaloids, most of which haven't really been evaluated in the past, and we're not exactly sure what they do, and it's a big question. This is why we prefer working with the purified chemical, which is, in our case, is a 99.5% pure. When Howard began production of Ibogaine, when Howard began Ibogaine development, they contacted a Belgian company, Omnichem, and Omnichem began the manufacture of GMP Ibogaine specifically for the clinical trial in Miami and for the National Institute of Drug Abuse, who purchased a kilo of the product from Omnichem. These are samples of the batch. Howard is developing Ibogaine under a trademark of Endabuse. These are the physical characteristics of Ibogaine base with its chemical formula, molecular weight, melting points. According to the literature, it's practically insoluble. However, we have been working on a new technology to develop higher yields and greater solubility. So the background, basically, it's used for hundreds of years in African medicine and religion. It was isolated by Dubinsky and Landrin in 1901. And in 1962, Lotsof discovered the anti-addictive effects. Naranjo, the Chilean psychiatrist, along with Leo Zeff, who was the subject of a recent book called The Secret Chief, worked with Ibogaine in psychotherapy and reported it in their work. In 1991, the National Institute of Drug Abuse initiated an evaluation of Ibogaine, and in 1993, the FDA approved clinical studies. The studies began in 1994 under Howard's tutelage, NDA's tutelage, in the Republic of Panama. We received permission from the government of Panama to administer this as an experimental therapy. We worked in several hospitals, and it was an absolute pleasure to work in a fully staffed, fully equipped hospital. The patients were in no way put in any discomfort. Rather, the opposite was true. They really adapted very, very well to a caring staff and an addict advocate, the presence of addict advocates, along with medical doctors and trained staff. Humatech began developing a protocol for a clinical study in Israel, and in 2004, the Israeli Ministry of Health approved a clinical study for Ibogaine. The problem was that there was no supply of GMP product. The manufacturer, Omnichem, had discontinued the product. A GMP product was formulated in Lithuania by a company named Melapharma, but they discontinued when Lithuania became a member of the European Union and raised their standards of what actually constitutes GMP material to internationally recognized standards rather than Lithuanian standards. So we were stuck with a approved clinical study with all of the things that are necessary that go along with it, the case report forms, the investigator's brochure, everything was in place. We were going to be working at a Israeli government hospital, Be'er Yaakov Medical Facility, and our principal investigator was the former chief psychiatrist of the Israeli Defense Forces and the head of psychiatric medicine at Ben Gurion University. So we had everything in place, but we didn't have a product. So I began an initiative to bring about a GMP product which could be used for clinical trials, but it wasn't as easy as we thought it would be. The National Institute of Drug Abuse conducted an enormous amount of preclinical work on Ibogaine and produced 16 volumes of data. And most of this data was ended up actually supporting all of the claims advanced by Howard Lotsof in his patent portfolio, which included that it was a broad-spectrum anti-addictive capable of treating narcotic addiction, cocaine and amphetamine abuse, alcohol dependency, nicotine dependency, and polydrug dependency. This action has never been seen before in any anti-addictive drug. In fact, pharmaceutical company ideology is based on the concept of, for every indication, there is a medication. And because we were a broad-spectrum anti-addictive, most of the science world initially rejected these claims as preposterous. Because how could a drug that's effective in treating opiate narcotics be similarly effective when treating cocaine or amphetamine or alcohol or nicotine? Because all of these things operate in different areas of the brain. And it was it was viewed with great skepticism, to say the least. However, it was later determined that Ibogaine does work in every one of these areas. Thereafter, we were referred to as a, quote, dirty drug, which means merely that it has multiple points of action. So we went into preclinical studies. Howard and Norma and I were part of a delegation to meet with the president of Gabon, who, when he, after a lot of discussions, decided that he would present us with emergency medical research supplies. He gave us 40 kilograms, lifted the embargo, permitted its airlifting to France, where it was then extracted and given to Dr. Jolix and Kaplan from the Erasmus University. And they published the first paper in the modern era of Ibogaine in the treatment of drug dependency. This was demonstrated that morphine-dependent rats who were given a naloxone challenge would have all of their symptoms basically eliminated. Additional work at Albany Medical College confirmed Jolich's work. Glick was the leading presenter. Then the papers started coming in from all over. And Rotterdam produced a study about the effects of cocaine, of Ibogaine on cocaine self-administration. Never before had a drug been seen capable of interrupting self-administration of cocaine. Rezvani at Skipper Bowles at Duke University says Ibogaine effects on alcohol were in reducing the amount of alcohol consumed dramatically in rats that were genetically designed to be alcohol preferring. And here's a slide indicating how much of a reduction there was. There were a lot of different studies that were done on Ibogaine that all confirmed the positions that were advanced by Howard in his initial work. Then came neurotoxicity questions. NIDA was busy developing other drugs like buprenorphine and LAM, which turned out to be a total disaster. And they decided that they were going to sabotage Ibogaine research. And what they did was they contracted with Johns Hopkins, with Mark Molliver, and he took a whole bunch of laboratory animals and gave them doses 10, 15, 20 times greater than we propose to give to humans. He failed to disclose in his paper how many of the animals died. The amounts he gave far exceeded the LD50 of these animals. But those that survived did suffer Purkinje cell loss. So then came the smear, the great lie. Ibogaine was neurotoxic. It's kind of like parquet margarine and butter. You say parquet, and they go, butter, butter, parquet. No, they would say Ibogaine, neurotoxic. So that was their approach until Zhu and others produced research demonstrating that there was no neurotoxicity at clinical doses. And Zhu was associated with Saeed Ali at the FDA laboratory, Toxicological Research, it's the National Center for Toxicological Research, down south. It's in Jefferson, Arkansas. And Saeed Ali is the principal in an organization called IDARS, which is the International Drug Researchers Association. And there are multiple mechanisms of action which are proposed. It appears to be working in a lot of different areas. And these systems define the pharmacology of human behavior affecting pain, pleasure, anxiety, depression, as well as neuron growth with inherent impact on memory and learning. And we move on to clinical studies. That NIDA funds 85% of drug addiction research throughout the world. And NIDA provided for us 16 volumes of data which can be used in support of a application for a new drug application, or an IND, an investigational new drug application. And the amount of material they have is vast. They include acute oral toxicity studies, range-finding studies, dose-response neurotoxicity studies in rats. There are studies in dogs, neurotoxicity studies in dogs, and so on. Then became the issue of Ibogaine deaths, and because there were a number of Ibogaine-related deaths, whereas the critics were quick to say another Ibogaine death has occurred, the review of the data and autopsies conducted, and all of the evidence indicates that they may have been related to Ibogaine, but Ibogaine was not the cause of death. In some cases, it was pre-existing cardiovascular issues that weren't properly screened. In other instances, it was the surreptitious use of drugs. But speaking about deaths, FDA-approved drugs administered in U.S. hospitals were reported at 112,000 last year, considerably more than so-called Ibogaine.
Isn't that correctly prescribed drugs?
Yes, those are correctly prescribed drugs, but sometimes they're over-prescribed. Sometimes they're given, the wrong drugs are administered. The nurse is pushing the cart down the hospital hall, and she gives one patient the other patient's drugs. Lots of stuff happens.
That's not included in that number. That's only drugs that were properly administered.
Properly administered. It may well be.
Mistakes are another number.
So we know that Ibogaine presents advantages over every other modality, that it significantly reduces withdrawal symptoms, it interrupts drug craving, it interrupts drug self-administration, it returns patients to their pre-addictive state, it eliminates the stigma of being an addict, and it returns the people to free choice, which is a very important issue because as former providers and present providers will know, that abstinence is not the goal. The goal is freedom of choice. So we have a situation where nothing does this as well as Ibogaine, yet we want to see Ibogaine developed as rapidly as possible so that people can go into the hospital, they can register, and they can detoxify with dignity and painlessly in a safe medical environment. And therefore, it is our approach to be very focused in dealing with the Food and Drug Administration. We are not seeking to address issues whether or not it interrupts drug craving, whether or not it interrupts self-administration, because it opens up a Pandora's box. How long does it work to interrupt self-administration? Well, some people for 2 months, some people for 6 months, some people for the rest of their lives, and some people for 2 days. We don't want to get into a boondoggle of studying this drug forever. We want FDA approval, and we want it the fastest way possible. And we have determined that the best way to get FDA approval and the best way to bring this medication to those who need it most is to concentrate on only one indication, the elimination of withdrawal symptoms, which we all know Ibogaine does so beautifully. So if we focus on that, well, from narcotic addiction withdrawal, that would be the indication, but everyone knows that no matter what a drug is approved for, off-label use is permitted. So therefore, once we're approved for this treatment for opioid withdrawal symptoms, then it can be used for cocaine, it can be used for alcohol, it can be used for all of the broad range of things that we want to do. So basically, Dr. Alper, who presented the other day, is the author of a number of papers about this subject, and I think we should move on to manufacturing, or if we're going to go, okay, Alpra studies reported that most of the people who were treated with Ibogaine were treated for opioid withdrawal. There was a greater percentage of people seeking that than any other method, although there were people who were seeking spiritual experiences and people who were seeking treatment for other indications as well. And so it was, as it was previously stated, over 5,000 people now we know have been treated with Ibogaine. These figures up to 4,900 only reflect to 2006. And we know that Ibogaine is growing at a rate of about 25% a year of patients being treated. We proposed this study in India, which was an open, single-dose, escalating, fixed-dose study of the safety and efficacy of Ibogaine in opioid-dependent patients. Our objectives of the study are to investigate the short-term effects of Ibogaine on opioid withdrawal, to investigate the safety of Ibogaine during acute withdrawal, an 8-week post-treatment evaluation, and to investigate the long-term effects on craving and substance abuse. And there are secondary questions which pop up too, and as well as pharmacokinetics. We have a very simple design. This is the study design that we want to use as the basis for an international multi-site study. The first phase consists of 6 to 10 days hospitalization. We stabilize the patient. We give them a comprehensive medical and psychiatric assessment. We take 4 volunteers who meet our criteria, and we administer a single therapeutic dose. After them, time is allotted for recovery, and then we go into the second phase, the follow-up. After they're discharged, they come back every week for a meeting with the medical staff and psychiatric staff. We have 8 weekly follow-ups scheduled to evaluate the long-term effects, the after effects, and what goes on in these people's lives and how they're redirecting themselves in what they're doing. So for safety reasons, the dose is escalated. We proposed that we start with 8 milligrams a kilogram and go to 12 milligrams a kilogram and then up to 16 milligrams a kilogram. This way, we can define what's the optimum dose response. So after the first group is done, which is going to take 2 to 3 months, but it's kind of like part-time. Most clinical work is very, very cumbersome and very time-consuming, and it goes on, and it's never-ending. You're doing studies for years, and they're the principal thing you're focused with. But this is very manageable study. We got 4 patients. They get treated. We follow them up, and then afterwards, 3 months later, we take in the next group, subject group B, and they get the second highest dose. And then finally, after their process, the data is captured and recorded. We can move on to the third subject, Group C. And this study offers a paradigm to the medical and psychiatric community and a unique opportunity to evaluate this new technology for the treatment of opioid dependence. And that's it for the clinical development. And we're still stuck without a product. We've got the study and we've got all the things, but we are working on the product. And I think, can I have some? Nope. Escape. Can I have some help? Which one changes?
Where's the other one? This one?
The second one.
This one?
That's it, yeah. Great. I just got back from my third trip to India. We have a contract manufacturing organization in Chennai, which is formerly Madras. It's in the south of India on the east coast. And we have begun a association with them, and we have an agreement for them to be producing Ibogaine for the next 10 years. We have decided to follow a particular route in the manufacture. We do not use Tabernanthe iboga. We use a similar plant from the same genus, which is a cousin or sister plant, Voacanja africana. We are using a technology that was developed by Omnichem. When Omnichem discontinued, we contacted Omnichem about obtaining their technology and the drug master file, which is on file with the FDA and specifically relates to the investigational new drug application for the University of Miami study. And it's important that when you're involved in clinical studies to use, if at all possible, the same product throughout every study, no matter where it is. Because if you start to do a study here and you're using one source of Ibogaine and they're doing another source in studies in Israel and they're using a third source for studies in another country, then you have to get involved with bioavailability studies. You have to involve yourself in a whole range of complications. We feel, and all people involved in pharmaceutical development feel, that the most direct way to regulatory development is to use the same product. So we acquired the manufacturing technology from Omnichem, which is contained in the DMF, and we're using the same formulation. We have, however, made great improvements. So we begin with the bark of Voacanja africana. They just completed the first processing of the first 250 kilograms made in non-GMP. This is the first production run. The second 250 kilograms will be processed to internationally recognized standards of good manufacturing practice and will be available for clinical trials. And the third run is for 500 kilograms. So altogether, our initial contract with them calls for the processing of 1 ton of botanical product. We've already acquired and have in India 50% of what we need. The preparation of ibogaine hydrochloride, it goes over from base into ibogaine hydrochloride crude, and then from the crude it's made into a pure product. And here's a rundown on the process flow. It starts out, of course, with the bark, and then comes the extraction and the concentrations of toluene and the pH adjustment and filtration and filtration of total alkaloids dissolution, and extraction in ethyl acetate. It continues on to you get a vocamine isolation, and a vocamine is like a total alkaloid, and then you bring it down from pure fractions and keep purifying it, and finally you come down to you get a first crop and a second crop, and then you end up with base. And then you have to turn the base into inorganic salts. And then you have to convert that to ibogaine. And the ibogaine that you get initially is crude ibogaine. It has to be recrystallized. It has to be reprocessed and made into pure. We're getting into improvements. Everybody's concerned about the scarcity of botanical material, especially Tabernanthe iboga, because there's been tremendous interest in the development of ibogaine, and people have been flocking to Cameroon and Gabon and scarfing it up as fast as they can, and without necessarily taking into consideration basic ecological principles, such as you take one plant, you have to plant two. So there's a shortage, and it's particularly felt in the people who are working with it, and also with the Africans, because there's fear that Tabernanthe iboga may become on the list of possible extinction if it's over developed, over harvested. One of the reasons we're working with Ibogaine is that there's Ibogaine africana is plentiful and it's cultivated, and we can provide a steady supply. When Omnichem was producing it, they had two runs, one in 91 and one in 94, and they were processing over 2 tons at a time. The first one was over 2 tons, the other one was 1,500 kilograms. But Omnichem was only able to get, well, it says 11%, but actually it's 0.0011%. They express it this way. And they had, the second one was disastrous for them. That was because they were getting an inferior product. Sometimes the alkaloid content always varies from plant to plant. And sometimes you get too much rain or not enough rain, and the alkaloid content varies from harvest to harvest. So we did some initial work with the Omnichem procedure in India with malady, and they were able to up it 2 points to 13%. But when we did this 250 kilogram production run, they raised the output by 3 times. They developed a improved technology, and the figures are staggering, because not only will we be able to produce more material, but we don't have to do as much harvesting in order to get this. Then, of course, it has to be brought from ibogaine base to ibogaine hydrochloride. Then it has to be made from crude to pure. This new process, they basically explain it in a process comparison, why they managed to get a increased output. And they worked very, very hard on this, and we were very pleased to see the type of yields. These are greatly increased yields that far beyond what we had originally expected. And here you can see all of the many steps that are involved of taking this product from the bark and ending up with, on the bottom, with a voacangine crop. And the traceability tree continues on to the manufacture of ibogaine hydrochloride. And this is not chemistry class stuff. This is very complex, done by a manufacturer of APIs, active pharmaceutical ingredients, and legitimate pharmaceutical drugs. And here's how basically they developed it, what they did, how they did it, the different batches. Go here. work is to be continued. They have different, now here's, we have to develop an analytical method which is required for regulatory purposes. And that in itself is a major undertaking. And, but we have a schedule, and we're working on that, the development of the analytical, but most important, we're working on the development of GMP batches, which is the goal of this entire exercise. And it looks like in May of this year, we'll have it available from 250 kilograms. And thank you very much. And I hope I can answer any questions you have.
Is your control of this process ironclad, or are we going to be seeing manufacturing going on in Miami next?
Well, it's not an easy thing to do. And because anybody who's interested in developing Ibogaine should be wanting to use the same product as was used in the FDA-approved clinical trial. After all, this manufacturing technology is on file with the FDA. You don't have, you know, we're of course going to amend it because we've made improvements and advances in yield and production, but I think that we're going to establish the gold standard of the product. This, by the way, is 99.5% pure. Nothing else approaches that. And so we think that, see, let me just address one point, you know, and that has to do with the risk versus benefit ratio and the administration of Ibogaine to human subjects. You know, addiction is a life-threatening illness. And if you want to interrupt that illness, if you want to effectively treat the illness, and there is no GMP Ibogaine available, because it doesn't exist, is the risk versus benefit ratio in favor of administering a drug of questionable origin to a human subject? And I, for one, believe that it is. That I have used material as a treatment provider, and I know that treatment providers are using material that is not GMP because it doesn't exist. They're using material of varying quality, of varying grades. Some are using total alkaloids and are finding results. Some are using people of material of 60, 70, 80% purity. Nobody wants to discuss with, no provider wants to discuss with other providers who their sources are, because everybody wants to keep it close to the vest. Everybody's getting material from different people. Some people getting it from Slovenia, some people getting it from Italy, some people getting it from Britain, some people are getting it from South Africa. So, you know, there needs to be a standardization of it. And so what happens when all of a sudden we come along and we have a product that's 99% pure in GMP? You see, what does this do to the other products, you know? And the only analogy I can make is that of prohibition, you see, because before prohibition in America, there were people who were making it, making wine, beer, alcohol, spirits, and then it was not available. So there was a need, there was a market that had to be filled. So people began to make it. And some of them made it in stills that they set up, and some of them made them in the makeshift factories, and some of them made it in a bathtub. And some of it was good hooch, and some of it would make you blind or kill you. But that was bootleg. But once prohibition was repealed and people began to make alcohol and spirits in specific proofs with a specific formulation and wines in a certain way, the need for bootleg material diminished. And we think that patients deserve to be provided a medication that was intended for human consumption. Material that is made in non-GMP is generally labeled by all reputable manufacturers as for research purposes only, not for human consumption. So, well, we're taking, providers are taking a risk, giving, especially in the area I feel of total alkaloids, because many cannot even tell you what those total alkaloids are, Well, what are the 12 alkaloids involved that you're administering to humans? So, so that's what I feel about GMP material, and I hope that people will think of it in a positive way. That because the most important thing for the provider should be the safety and comfort of the patient, and not how much money they're making or how much costs a gram of Ibogaine or how much is this or how much is that, because the cost of that gram of Ibogaine is comparatively small when you compare it to the cost of taking care of somebody for 7 to 14 days in a recovery situation. So let's move up to a better product. Yes.
The actual Iboga and the Ibogaine, what's the difference in that? The plant itself, using that as opposed to using hydrochloric acid and the different processes?
Well, the plant is the plant. It's the difference between coca and cocaine. A plant contains many alkaloids, and in order to ingest the equivocal amount of Ibogaine, say you were taking a gram of Ibogaine, giving a patient a gram of Ibogaine for treatment, in Africa, in order to get that amount of Ibogaine, they have to consume almost a kilogram of ground botanical, and it's an all-night affair. They feed it to people with a spoon. They eat it. They vomit. They eat it. They vomit. They dance. But what you're getting is the pure alkaloid. You're getting without any of the impurities. This is a pharmaceutical grade product that's manufactured to standards for intended for human consumption.
The reason I asked,
Yes.
sorry to take the time up. When I was in Africa, the people who actually used it didn't go back on drugs, and the people who used Ibogaine, some of them previous habits or environmental conditions. So the message from Africa was to the people making it here, shouldn't they make Ibogaine and then use that with some Iboga, crushed Iboga?
Well, that requires a lot of testing. As Deborah Mash once told me, in God we trust, others show data. So if
But isn't what Bob is proposing, that's not saying that research doesn't need to be done. He's looking for the fastest way to get the best, purest, most Western standard compound to market. And then worry about comparing them later when you can actually make
It's illegal right
scientific
now.
comparisons, not just, oh, the total extract is better. That's, a lot of people are saying that, but then we have to figure out what's causing that belief.
See, right now, Ibogaine is available in boutique settings throughout the world. There are boutique clinics operated by dedicated, committed people, many of whom are recovered addicts and who have had their lives turned around. And I think it's wonderful, you see, but there are only a certain amount of people who can be treated in these type clinics. And what I'm really striving for is for somebody to be able to go into Beth Israel, or go into Harlem Hospital, or go into a facility. They may have to sign up and go on a waiting list, you see, but to be able to have a hospital bed for a week for a clinical detoxification in a safe environment and the medical supervision, you have the ability to treat thousands upon thousands of people this way. And I'm sure that boutique treatment providers will continue to operate and will continue to attract those people who would prefer to go down in a situation like that rather than going to a hospital. But there are many, many people who are on public assistance, who have Medicaid and things like this, who have no way to pay to go to Mexico and no way to pay to go to Vancouver or some other place to do it. And I know that these people are also treating people for nothing in many instances or at reduced rates. And I understand it's a sliding scale, but there are people on the bottom. Don't forget, the people we're intending this product for occupy the lowest rung on the social ladder, you know. And while there are well-off addicts, you know, the majority are not. And we'd like to see it available in a hospital. And this is the way we have to approach this, by providing a GMP product that will be acceptable to the FDA and others. Yes?
Well, I'm sure no one here would argue against making Ibogaine more and more legitimate.
Mm-hmm.
But there's some really basic things that have not been done to Ibogaine that are done to every other drug.
Mm-hmm.
And I actually was bothered by one of your earliest slides that showed the Ibogaine receptor. And it had a bunch of different receptors up there.
Mm-hmm.
That doesn't seem likely to me. It seems that the Ibogaine receptor has not yet been nailed down. So how do we approach, you know, more scientific credibility?
Well, mechanism of action is not required by the FDA. We don't have to demonstrate mechanism of action. There are some people who say, well, it's a result of it being sequestered in the fat and released slowly and this accounts for the after effects. Other people have other hypotheses that they put forth about mechanism of action. But it's really a tempest in a teapot, because the FDA requires only two criteria be met: a demonstration of safety and one of efficacy. And you know, it's even questionable whether we know about what's the mechanism of action of aspirin. I'm sure somebody might have figured that one out by now. But
Are the only drug that all people identify as working
yes.
is morphine or opium derivatives?
Mm.
Is historically the only drug where all doctors and all researchers agree
Mm-hmm.
works.
It works.
Human history.
Well, pardon me? Okay. Well, thank you very much for your attention, and